In the soft hush of Earth's earliest oceans, before bones, before blood, before breath, something remarkable begins, not with a flash, not with a sound, but with a film. Across the surface of a droplet, lipids arrange themselves not by choice but by tendency. Tails tucked inward, heads turned outward like petals around a secret. These are not walls, not yet only boundaries, delicate and trembling, separating nothing from something. A membrane forms not to defend, but to hold. It is the simplest architecture of life, a closed space where the outside slows down and the inside starts to mean something. No blueprint guides it. No command issues the order, just molecules settling where they prefer to be, obeying only the physics of their own quiet repulsion and attraction. In this sealed droplet, the water inside grows distinct from the water outside. Salt concentrations diverge. A difference is born, and with it the seed of all that will come. The first protocell drifts through a lukewarm tide, unknowing and alone. It does not feed. It does not know it exists. But its boundary, its membrane, is already a marvel of quiet constraint. The amphiphilic lipids that form it are not rare, not magical. They self-assemble when the conditions allow, like dancers finding one another by rhythm alone. Once closed, this membrane does something strange. It traps. It captures molecules inside, not with intent, but with inevitability. Perhaps a catalyst lingers here, a molecule that under the membrane's shelter begins to fold proteins from raw peptides or nudge sugars into rings. Reactions inside become just a little more likely than those outside. It's a small difference. But lifelike erosion is a patient art of accumulation. Within this primitive pocket, no nucleus waits, no organelles stir. There is only fluid enclosed. But even fluid carries motion. Brownian jitter nudges molecules into collisions. Some react, others float inert. An ancient irony, life begins not with precision, but with chaos filtered through boundaries. What persists, persists. What dissolves, vanishes. No audience observes, no mind records. And yet something real is taking shape. The membrane, though supple, is not passive. It curves it, flexes it, adjusts its permeability. Some molecules cross freely water-oxygen, perhaps ammonia. Others cannot pass without help. Channels form rudimentary pores sculpted by accident or chemical luck. In some places, proteins embed themselves into the lipid sea, creating portals through which charged ions can drift. These are the first gates, the first choices, though the cell does not choose. Outside, the ocean remains vast and indifferent. Volcanic plumes spew heat and metals into the water column. Ultraviolet light sears the surface. The sky is hostile, but within the membrane there is buffering. Reactions proceed in gentler gradients. A different world begins to flicker, not safer, not simpler, just separated. The cell is not alive in the way we think of it. It does not divide yet. It does not reproduce. But it has direction, a bias toward internal order that the open sea cannot match. It is easy to miss the irony. Life begins by stepping back, not forward, by hiding, by walling off a space too small to see. And the membrane, that modest envelope, becomes the first architecture of purpose. Not intelligence, not desire, just the slow pressure of physics reshaping chemistry into systems that persist. Boundaries once formed change everything. They enable difference, they hold gradients, they nurture imbalance, and from imbalance flow begins. The inner soup, if it can be called that, is not a single substance, but a tangled dance of organics, amino acids drifting past catalytic fragments, simple sugars colliding with phosphate chains. These components do not yet form a genome, but a logic is stirring not in letters but in behavior. Molecules that replicate imperfectly begin to outlast those that don't. Chains that fold into stability remain longer than those that unravel. There is no competition, only consequence. No strategy, only structure. And somehow this is enough. The membrane grows, taking in lipids from its surroundings. It flexes with heat, stiffens in cold, becomes more or less porous, depending on the ions that nudge against it. It is not inert, it is dynamic, a quiet marvel of self-adjustment. When the cell absorbs too much, it may rupture, spilling its contents into the sea, starting again. But sometimes, in just the right moment, it divides. Not cleanly, not with symmetry, just a stretching, a pinching, a break. Two cells where once was one. Not identity, not inheritance, just continuity, like a ripple breaking into two. This division is not yet reproduction. It is copying without memory, separation without intent. But even this crude motion lays the groundwork for something else. Because with each new boundary comes a chance to trap a new combination inside. And some combinations prove more persistent, more capable of sustaining reactions that extend the life of the cell just a little longer. Not better, just longer. And in deep time, longer becomes everything. No fossil records this moment. No rock remembers the cell's first enclosure. But the principles echo forward. Every cell that exists today in fern, in fungus, in whale and bird and human, carries a membrane descended from these first films. More complex now, elaborately studied with proteins, sugars, sensors. But the principle remains a boundary that separates, sustains and responds. There is a quiet mystery here. Why should certain molecules arrange themselves into enclosures at all? Why should enclosed chemistry outperform open reaction? The answer is partial. A membrane allows for gradients differences in pH, in charge, in concentration, and gradients are energy. But why should life choose the path of compartments over chaos? We do not know. We may never fully know. Perhaps because once enclosed molecules linger. They form longer chains. They are not swept away. Perhaps that pause, that brief shelter, is all that was ever needed. Only took a few million years to fold a film. Only took a billion more to make one that could remember. Only took the quiet weight of time pressing against nothing until something yielded. The boundary once made begins a lineage that does not break. All that will follow, growth, memory, division, pain, love, will require this first act, the quiet making of a space apart from the world. And so the first cell does not speak, does not stir the tide. It floats sealed. But inside the conditions shift. Reactions begin to loop. Catalysts begin to repeat. And life, if it can be called that, takes its first organized breath, even if no air is yet involved. Long before thought, long before neurons or blood, there are gradients, quiet, invisible slopes drawn across the chemistry of early oceans. Where hot meets cold, where acid meets base, where salt meets fresh, the world begins to hum with imbalance. These gradients are not seen but felt by molecules coaxing them into motion. A proton here, an ion there, drifting not randomly, but along paths made subtly downhill by energy's soft insistence. The earliest membranes do not seal completely. They leak, they flutter, they permit passage in ways that would seem sloppy by modern standards. But they allow just enough selectivity for the outside and the inside to differ, and in that difference a strange new order begins to grow. The cell becomes a place of potential, its inside not only separate but charged. On one side of the membrane more hydrogen ions gather, on the other fewer. The imbalance is not a flaw, it is a force. Energy in these early moments does not roar or spark, it diffuses. This cell is powered not by flames but by trickles, proton gradients, slipping through protein gates driving rotary enzymes like molecular water wheels. ATP synthase, one of the oldest molecular machines in biology, begins to turn. It is not constructed with foresight. It is not designed. but its spiral shaft rotates binding ADP and phosphate into ATP, the basic unit of energy that all life still uses. A slow twist, a binding click, and a high-energy bond is formed. This process is almost miraculous in its persistence. Across all known life, from the simplest bacteria to the tallest tree, this rotary motor remains. Its core mechanism has not been improved upon, only inherited. The early cell nestled in the dark currents of the Archean Sea becomes a factory of imbalance. Sodium outside, potassium within. Calcium spikes, chloride dips. Not chaos, but order made from unevenness. Like a battery, the cell holds charge across its membrane. And like a battery, it can spend that charge to do work, importing nutrients, exporting waste, sending signals that do not yet rise to the level of communication. This flow, slow, subtle, directional, is the beginning of agency, if not intention. Life begins not with a plan, but with a slope and the means to follow it. The environment outside is anything but static. Hydrothermal vents erupt, releasing metal ions and reducing gases. Sunlight penetrates only the shallowest layers filtered through haze. But beneath it all, gradients persist. Some are thermal, with warmth radiating from the crust. Others are chemical, the result of ancient rock and water exchanging their gifts. Some cells drift closest to vents where energy is plentiful but conditions harsh. Others stay in gentler zones where gradients are weaker but survival more likely. A soft sorting begins not selection by struggle but divergence by preference. No will governs these movements, only tendencies, only thresholds. It is tempting to imagine these cells as striving, as seeking better gradients, but they are not seekers, they are responders. Their proteins open or close in the presence of specific ions. Their membranes flex or harden depending on fatty acid length. No awareness exists, yet still responses occur. Still the environment shapes what persists, and what persists begins to leave echoes. Some gradients arise not from the world but from within the cell itself. As reactions proceed, hydrogen ions accumulate, lowering internal pH. The cell adjusts, exporting excess protons to maintain balance. Not consciously, not optimally, but sufficiently. This homeostasis, the quiet maintenance of internal conditions, becomes one of life's central themes. It is not dramatic, it is not visible, but it is relentless. Life, it seems, is less about change than about staying the same in a changing world. Here too lies irony. The earlier signs of organization do not come from control but from constraint. A cell survives not by shaping the ocean, but by shaping its response to the ocean. It builds pumps to push against gradients, even as it depends on other gradients to power those very pumps. This circular logic dependence on imbalance to preserve balance defines biological life. It is not solved. It is managed. No fossil records these gradients. No stone holds the memory of a proton's path across a membrane, but their legacy endures in every heartbeat, every synapse, every breath. Nerve cells fire by altering ion gradients. Muscles contract by releasing calcium. Mitochondria function by passing electrons down gradient-driven chains. The subtle physics of the early ocean becomes the framework for complex behaviors billions of years later. There are questions we still cannot answer. Why did some cells evolve more efficient pumps? Why did others lose the ability to generate their own gradients and instead parasitize their neighbors? Why in some lineages did membranes grow thick and regulated, while others remained porous and fragile? These are not questions with simple answers. Perhaps the conditions varied. Perhaps the accidents accumulated. Perhaps the gradients themselves selected for complexity. The truth is buried in deep time, washed away by tides no longer traceable. Only took a few nanometers of membrane to divide inside from out. Only took a few millivolts of potential to power the chemistry of life. only took the slow drift of charged particles through a protein's pore to spin the first motor. Life begins not with leaps, but with leaks, not with clarity, but with concentration. The cell, though alone, is never isolated. It sits in an ocean of gradients of temperature, salinity, light. Each one pushes and pulls, encouraging one pathway, discouraging another. Over time, those that align with the drift persist. Those that don't dissolve. In this way, a system emerges not through competition but through compatibility. The cell's internal order reflects its external environment, shaped less by ambition than by resonance. Eventually, some cells begin to generate their own gradients, exporting ions with more regularity, capturing energy from redox reactions, layering membranes for efficiency. But even in these refinements, the principle remains the same. Life is a conversation between inside and out, and gradients are its quiet language. No sound, no signal, no intent. Just differences patiently exploited. The ocean rolls on unaware. Cells multiply. Some die. Others persist. None remember. None plan. But in their chemistry, a logic begins to hum, a logic of flow, of resistance, of return. And through it all, the gradients remain invisible, essential, and quietly organizing the earliest whispers of life. It is not the shape of the cell that matters, not its smallness or its impermanence, but what it begins to contain. Inside this boundary of lipids and gradients, the internal world thickens. Reactions do not occur everywhere all at once. Instead, they localize, concentrate, and settle into patterns. Ribozymes twist into folded scaffolds, catalyzing reactions with no consciousness, only configuration. Chains of amino acids begin to form, coiling into the first fragile enzymes. Some break apart sugars. Others assemble nucleotides. Still others help to stabilize the very membranes that shelter them. There is no director, no center. Yet already the cell shows a tendency to organize from within, not with intention, but with persistence. Chemistry, when enclosed, becomes choreography. Molecules encounter each other not as rare accidents, but as expected possibilities. The interior of the cell becomes thick with potential, a liquid tangle where chance meets constraint. Within this cytoplasmic soup, the beginnings of metabolism arise, simple loops of reaction, each step nudging the next, not because it should, but because energy allows it. These are not yet the grand cycles of the modern cell. No Krebs, no Calvin, no elegance, just sequences that lead somewhere useful before degrading into waste. Molecules are activated, transformed, recycled. Some reactions produce byproducts that alter the pH, shifting the internal balance. Others release energy, and that energy in turn powers the continuation of the loop. Life begins not with structure, but with feedback. The cell becomes not only a container of chemistry, but a maintainer of it. To persist is to balance gently and always between too much and too little. At the heart of this early metabolism lies the problem of specificity. Reactions must happen, but not just any reactions. The wrong combination could destroy the cell from the inside. So selection begins not by choice, but by stability. Enzymes that catalyze safe, repeatable steps tend to be preserved. Molecules that bind too strongly or not at all are washed away. Over generations, only those patterns that coexist without collapsing are passed on. There is no memory here, no genome yet, just continuity through compatibility. Only took a few stable loops to define the earliest metabolism, only took a few 100 million years to refine them. The cell's interior is not homogeneous. Some regions become denser with activity, others quieter. Proteins float and cluster not at random but with preference. Hydrophobic domains avoid water nestling into each other. Charged regions interact across the cytoplasm like distant echoes. Order begins to emerge from proximity and charge, not symmetry, not design. just the subtle shaping of space by molecular need. In these gentle asymmetries, compartments begin to hint at themselves. Not true organelles, not yet, but distinctions in local function. One area richer in synthesis, another richer in energy transfer. These are not built, they emerge. There is a softness to this process, a delicacy. Nothing is permanent. Bonds form break reform. The entire cell is fluid, not just its membrane, but its logic. And yet this fluidity is not chaos. It is adaptability. When conditions change, pathways shift. When the environment cools, reactions slow, but they do not halt. When nutrients dwindle, the cell retools, turning inward to recycle its own components. This capacity to shift without falling apart becomes one of life's defining principles. Not resilience through rigidity, but through liquidity. Perhaps the most astonishing feature of this early interior is its lack of hierarchy. No molecule is more important than another. No structure oversees the rest. Yet patterns hold. Cycles continue. Order persists. This decentralization, this self-regulation becomes the blueprint for all that will follow. Even in complex organisms, no single part controls the whole. Life remains a negotiation, a web of mutual dependency held together not by command but by chemistry. The cell's interior does more than maintain itself. It begins ever so slightly to shape its own evolution. Molecules that aid in replication of proteins, of ribozymes, of membrane components, begin to dominate. Not because they are better, but because they leave more traces behind. Persistence becomes its own reward. A molecule that helps the cell grow will by default become more common. No plan exists, no foresight. But already self-reinforcing patterns begin to spiral outward. It is easy to imagine progress here, but the truth is slower, more meandering. Most early cells fail. Their interiors collapse, or their membranes burst, or their chemistry veers into instability. These failures are not recorded, but the few that persist, that keep the delicate dance going, become the ancestors of everything to come. In their interiors we see not success, but continuation, not advancement, but endurance. There are mysteries still. How did the first enzymes achieve catalytic precision without error-checking machinery? How were harmful byproducts managed without compartmentalization? Why did certain metabolic loops stabilize while others failed? These questions linger not for lack of theories, but for lack of fossils. The interior of a cell does not fossilize. It dissolves, leaves no mark. We know it only through inference, through the echoes it leaves in the biochemistry of its descendants. And yet a few facts anchor us. All modern cells from archaea to mammals share certain metabolic cores: glycolysis, ATP synthesis, membrane potential. These commonalities suggest that the basic interior logic of cells stabilized early and has been maintained ever since. It was not perfected. It was simply good enough not to fall apart. And in biology, sometimes that is all that matters. Only took a lipid sphere to house it all. Only took a handful of reactions to generate usable energy. Only took the soft refusal to dissolve again and again for life to begin building from within. The cell does not need to be complex to be complete. Its order is not a product of size, but of slowness. And so inside this tiny boundary order begins not with rules, but with rhythm, a rhythm of reactions, of flows, of rising and falling concentrations. The cell breathes without lungs, thinks without mind, endures without memory. In its stillness, life becomes not an entity, but a space where chemistry folds into habit, where repetition gives rise to persistence, and where persistence becomes the quiet foundation of all that will follow. Somewhere in the silent interior of these early cells, coiled within the fluid and motionless to the eye lie strands of nucleotides, sequences of adenine, uracil, cytosine, and guanine drifting in a watery tangle. They do not look like meaning. They do not carry structure in the usual sense, and yet in their particular arrangement a new possibility begins to stir the ability not only to react but to remember. Ribosomes, those ancient RNA strands that both store information and catalyze reactions, float among the other molecules like quiet architects. Their shapes are not simple lines but complex folds, origami built from attraction and avoidance loops and stems twisted by base pairing. One fold binds a specific amino acid, another cuts a sequence at just the right point. These are not thoughts. They are physical coincidences that happen to persist. And in persistence, something larger takes shape, a system in which information guides transformation. The code has no author. It is not written, not intended. But it can copy itself. And in doing so, it begins to pass on a molecular memory. This memory is not flawless. Each replication brings errors. Bases are misread, skipped, inserted. But the cell does not care. It cannot. It has no self, no goals. What matters is that the strands that are copied even imperfectly outlast those that are not. Over time, mutations accumulate. Some break the molecule's ability to fold. Others make it fold better. Most do nothing at all. But in the slow drift of time, some variants prove more efficient at catalysis, at stability, at attracting complementary sequences. And so quietly, without intent, the sequences that persist begin to dominate. The cell does not evolve, its molecules do. And because they do, the cell endures. RNA is fragile. It breaks easily in alkaline environments. It hydrolyzes under heat. Yet here it is nestled in the protective interior of a membrane doing something no other molecule has done, acting as both template and tool. The early cell becomes a space where information can be stored and reused. Not just once, but again and again. And with reuse comes selection. And with selection, a soft memory begins to hum at the core of life. Not consciousness. Not even awareness, just the statistical echo of what worked yesterday shaping what is likely to remain tomorrow. As sequences grow longer, their possibilities multiply. Some ribozymes begin to catalyze the linking of amino acids. Peptides form short chains at first, then longer ones twisting into folds of their own. These peptides do not replace RNA, but they assist it. Some stabilize membranes. Others help with catalysis. A new kind of partnership emerges, one that still exists in every modern cell: nucleic acids, guiding protein formation, proteins supporting nucleic acids in return. This reciprocity, this division of labor is not designed. It simply proves persistent. And in biology, persistence is the only test that matters. At some point, perhaps slowly, perhaps all at once, a rudimentary code is born. Certain RNA sequences begin to attract specific amino acids. Base triplets, three-letter codons begin to correspond not perfectly, but reliably, to particular peptide fragments. These codons do not yet form a language, but they begin to carry consequence. A change in sequence can now change the structure of a resulting peptide. Information for the first time has a phenotype. The code is not universal, not yet, but it is directionally stable, and that stability over time allows complexity to accumulate. Only took a few base pairs to start linking acids into strands. Only took a few lucky folds to scaffold new reactions. Only took the passage of time soaked in silence for chemistry to become protocol. The modern genetic code, the mapping of 64 codons to 20 amino acids, is nearly universal today from bacteria to whales, but its origins are shrouded in uncertainty. Why these codons? Why these amino acids were others tried and discarded? Likely, yes. But no fossils record failed codes. Only the winners remain. The code we now use may not be optimal. It may simply be the one that worked well enough to outcompete its siblings, a soft tyranny of the probable. Even today, the genetic code is not always rigid. Some organisms deviate using different start codons, different assignments, but the deviations are minor conservative. The core logic holds. Once information begins to influence structure in a heritable way, the system becomes resistant to change, not because it cannot change, but because every mutation risks disrupting something already functional. In this way, biology begins to protect its own history. The cell becomes a library that edits itself slowly, cautiously preserving old notes even as it writes new ones. Evolution becomes less about innovation and more about the slow rearrangement of what already works. And yet, for all its elegance, the code remains blind. It does not understand itself. It does not choose what to remember. Errors are not mistakes in the moral sense. They are just biochemical events, mispairings, strand slips, oxidative damage. But some of these errors improbably lead to improvement, and so without knowing the system learns. There are mysteries here still unresolved. How did RNA first become self-replicating? How did base pairing evolve to be specific enough for fidelity, but loose enough to allow variation? Could alternative codes have supported life differently? These are not questions with tidy answers. They are questions that gesture toward the contingency of everything we know. The code that remembers could have remembered differently, and so could we. Today, every living cell still uses this code, not just similar codes, the same one, the same triplets, the same amino acids. This is one of the most astonishing facts in biology, that the memory first whispered in ancient oceans still whispers in the muscles of a whale, in the leaves of a tree, in the breath of a sleeping human. The code has travelled unchanged in essence through billions of years and across every branch of life. It did not have to, but once it took hold it held tightly. Only took a strand of 4 bases to begin it. Only took a fold that could replicate. Only took the quiet filtering of time, letting only the stable remain. The code is not a story. It is not a message. It is not even a decision. It is a chemical consequence, persistent, replicable, just unlikely enough to be rare and just likely enough to be everywhere. Inside every cell, the code continues to copy itself. With each generation, with each breath, with each wound, and healing it loops through its old sequence again, adding nothing new except the possibility of error. And somehow through that error, through that subtle forgetting and imperfect repetition, life continues to organize itself. There is a moment when the cell, having gathered its gradients, folded its catalysts, and encoded a simple memory in strands of nucleotides, begins to stretch. It is not dramatic. It does not split like a cracking stone or tear like muscle. Instead, it elongates slowly, patiently, as if testing the limits of its membrane. Inside, the contents swirl and distribute, some strands of RNA settling near the center, others drifting to the edges. Proteins orient along the inner surface, not in rows or patterns, but in enough symmetry to matter. Then, as pressure builds from growth or energy imbalance, the membrane pinches, not with control, not with precision, but with just enough tension to fold inward, deepen and close. One cell becomes 2, not by strategy, not by command, but by the physics of persistence. This is not reproduction in the sense we now know it. There is no sex, no chromosome, no mitotic choreography. It is division by necessity, compelled by surface area and diffusion limits, by the slow burden of becoming too full. The daughter cells are not identical. They do not inherit perfectly. They carry fragments of what came before, copies of RNA bits of catalyst residual gradients, and yet against the odds they function. This act of division is not a beginning. It is a continuation, a soft duplication of presence. The cell does not know it has split. It simply continues. But now instead of one reaction space there are two. Instead of one chance to persist there are two. And with each subsequent split life spreads not with conquest but with quiet redundancy. Cells do not grow to dominate. They grow because they can. In time, those that divide more evenly, that distribute their contents more reliably, tend to persist. Not because they are better, but because they are less likely to collapse. And so slowly mechanisms emerge to guide this process. Scaffolds form beneath the membrane. Anchor points hold strands in place. Gradients are reset post-division. Not flawlessly, not consciously, just often enough. Over millions of years, primitive division becomes refined. Cytoskeletal elements, primitive tubules or actin-like filaments begin to assist in the distribution of cellular contents. These are not yet true mitotic spindles, not yet mechanical systems, but early frameworks that help orient the chaos. Eventually molecules appear that detect membrane curvature, that initiate pinching when thresholds are reached. Still, there is no central planner, only feedback. The more organized the division, the more likely the progeny survive. And survival, though blind, shapes what remains. In this system, memory is imperfect. Errors occur. Some cells receive too few ribozymes. Others inherit malformed peptides. Most die. But death is silent. It leaves no trace in deep time. What remains again and again are those cells that divide just well enough. There is no perfection here, only tolerance. Division is not designed to be precise, it evolves toward being good enough. And sometimes just good enough becomes a principle. With division comes a new challenge, the problem of distribution. Resources must be shared, but not necessarily equally. The cell must balance competing needs, catalysts, information. Strands, membrane lipids, energy gradients. Some molecules are abundant, others rare. The stochastic nature of inheritance means that each division introduces variance. Some cells flourish, others do not. This variation, though unintentional, becomes the substrate of natural selection. And so slowly traits that promote equitable partitioning, such as tethering of key RNA sequences to scaffold proteins, begin to spread. Not because they are understood, but because they are not lost. It is one of the quiet ironies of biology. To persist, life must let go. Division means separation. It means risk. It means that whatever internal order a cell achieves must be capable of being broken apart and reassembled. There is no permanence, only pattern, only the soft rhythm of growth and halving like waves rising and breaking across a cellular shore. And with each cycle the potential for novelty increases, not planned novelty, emergent novelty, a strand of RNA that mutates just slightly, a protein fold that finds new stability, a membrane composition that better tolerates heat. These are not choices. They are consequences of division, of the simple fact that copying is never exact. In time, some lineages of cells begin to coordinate their division with environmental cues. When nutrients are scarce, they pause. When conditions improve, they resume. This regulation, though primitive, is a step toward timing, a step toward rhythm. Perhaps the beginnings of cycles, day and night, feast and famine. The cell becomes not only a container of reactions, but a participant in seasonal change. And in doing so, it begins to entangle itself with the larger patterns of the planet. There are mysteries here too. How did early cells manage asymmetrical division? Why did some lineages evolve toward regulated cycles while others did not? What were the constraints that shaped the first stable reproduction strategies? We have fragments of answers, modern analogues, theoretical models, but no fossils of these first divisions. We know only their implications, not their details. We know that modern cells divide with exquisite choreography, with spindle fibers and checkpoints, with regulated gene expression, and compartmental tracking. But these refinements came later. The earliest divisions were messy, inefficient, fragile, and yet they worked. Only took a pressure differential to start the pinch. Only took a fold in the membrane to split the volume. Only took the biased inheritance of a few strands to spark selection. The act of division, so mundane, so repetitive, is also foundational. Without it, there is no persistence. Without it, life would be a single cell drifting until it decayed. But with it, life can spread. Not outward in ambition, but inward in redundancy. Division is not expansion. It is repetition. And in repetition, patterns take hold. To divide is also to remain. A part of the old cell persists in the new. A loop of memory, a rhythm of reactions, a structure of lipids. Nothing complete is transferred, but nothing complete is lost. The cell becomes a relay, passing on not identity but instruction. Not essence, but process. And that process repeated again and again becomes the fabric of life. Not because it is efficient, not because it is meaningful, but because it works. Quietly, continuously, endlessly. In a world without intention, without correction, and without supervision, the first cells persist by embracing imperfection. Nothing inside them unfolds with absolute fidelity. RNA strands mispair, enzymes catalyze off-target reactions, membranes admit unwanted ions, and division distributes resources unevenly. Yet despite the accumulating error, or perhaps because of it, life does not collapse. It diversifies. The cell is not a fortress of precision. It is a compromise, a structure defined as much by what goes wrong as by what goes right. Errors are not punished. They are tested, filtered gently over generations, and slowly patterns emerge not despite the mistakes, but because of them. Every process inside the early cell is prone to failure. Catalytic folds sometimes destabilize. Replicated strands carry mismatched bases. Entire loops of metabolism stall because a single step is blocked by heat, acidity, or scarcity. There are no repair mechanisms, yet no proofreading polymerases, no error-correcting feedback loops. And yet across billions of replications, something astonishing happens. A system emerges that can tolerate and sometimes even benefit from being wrong. The cell becomes not a perfect replicator, but a stable experiment, a space where error rates are not eliminated but managed. Consider the copying of an RNA strand. Each nucleotide added carries a chance of substitution. A different base slips into place, changing the sequence downstream. Most of these errors are inconsequential. Some are lethal, but a rare few, alter the molecule in a way that makes it fold more stably, catalyze more efficiently, or resist degradation. These molecules outlast the others not because they were selected for, but because they happen to persist. Selection at this stage is not about advantage. It is about retention. The cell holds on to what doesn't fall apart. Over time, the tolerance for error becomes a quiet rule. Cells that are too precise miss opportunities to adapt. Cells that are too error-prone collapse into noise. Somewhere in between lies a narrow band of survivable imperfection, a corridor through which life flows. This corridor becomes self-reinforcing. Molecules that function within it help build environments where others can do the same. A primitive homeostasis forms not from conscious regulation, but from mutual stability. One molecule buffers the pH, another traps free radicals, a third anchors strands in place. Each contributes to keeping the system within functional bounds even when individual reactions fail. Ironically, the very instability of RNA may have accelerated life's persistence. Its susceptibility to mutation creates variety. Its simplicity allows it to replicate without proteins. Its fragility makes it temporary, a feature that favours flexibility over permanence. DNA will come later, offering stability and fidelity. But for now it is error that keeps life moving. Not cleanly, not efficiently, but sufficiently. In some lineages, rudimentary correction begins to emerge. RNA strands that fold in particular ways begin to exclude incorrect pairings. Some catalytic peptides enhance replication accuracy by stabilizing complementary base pairing. These mechanisms are not perfect, they only reduce the error rate slightly. But that slight reduction makes complex molecules more likely to persist. Complexity, then, is not the result of perfection, but of tolerable imperfection. The cell, in effect, learns to live with its mistakes, allowing just enough error to explore new configurations without unraveling completely. This learning is not conscious. It is chemical, it is statistical, and yet its effects are profound. With every generation, the cell's internal processes become a little more reliable, a little more robust. Feedback loops begin to form not as circuits but as consequence chains, one molecule's failure altering the abundance of another. Those loops that collapse are lost, those that hold are reinforced. Eventually, entire pathways emerge stable enough to persist through time, flexible enough to absorb shock. There are questions that remain unanswered. Why did RNA survive long enough to encode repair? Why did certain error rates become stable while others vanished? Did some early systems collapse because they were too accurate, too rigid to evolve? These questions are not merely theoretical. They strike at the heart of why life looks the way it does. Our genetic code, our metabolism, our capacity to adapt all rest on an ancient compromise between order and chaos, between the need to preserve and the need to change. Even today, errors persist at every level of biology. DNA replication, though remarkably accurate, still miscopies a few bases with each division. RNA transcription introduces mismatches. Proteins misfold. Some cells accumulate enough damage to die, others become dysfunctional, but the system continues. Redundancy buffers loss, plasticity absorbs damage, repair corrects what it can, and evolution discards what it cannot. The quiet rules of error remain embedded in the deepest layers of biological order. They are not flaws, they are the scaffolding of flexibility. Only took a misplaced base to reshape a molecule. Only took a single fold to open a new catalytic pathway. Only took the willingness of chemistry to continue even in the presence of noise, for life to become self-correcting without ever becoming perfect. Error is not the opposite of order, it is its companion. The two coexist in every cell, each holding the other in tension. And from that tension arises novelty, persistence, and the slow, unsteady march of biological time. Inside the earlier cells, none of this is apparent. There is no awareness of mutation, no concept of fidelity, only a system that endures because it does not collapse, because it is built to fail slightly repeatedly forever. And each failure is also a search, a chemical gamble, that in the vast slowness of deep time occasionally yields something that works just a little better than what came before. Life then does not seek perfection, it seeks continuity, not the avoidance of error but the capacity to remain through it. Somewhere in the warm shallows or near the mineral-rich mouths of submarine vents, single cells drift near one another not by will but by currents, not by purpose but by proximity. There is no touch of community here, no mind to grasp connection, and yet where cells gather something changes. Reactions spill beyond boundaries. Metabolites diffuse into shared space. Waste for one becomes food for another. No agreement is made, but mutual benefit arises silent and unacknowledged. This is not yet multicellularity. It is not even organized cooperation. but it is coexistence in which the survival of one cell becomes more likely when others linger nearby. The gradients between them begin to shape the direction of molecules drawing nutrients in shared flows, forming microenvironments where survival is less random, less harsh. These cellular gatherings are not structured yet. Over time, clusters form more frequently held together by surface charges, biofilms or extracellular scaffolds, primitive meshes of polysaccharides that begin to tether one cell gently to another. Within these clusters, something unexpected emerges: differentiation. A few cells begin to focus on producing specific molecules, enzymes, that break down tough nutrients or pigments that absorb light more effectively. Not because they are instructed to, not because they are less capable of doing other things, but because the presence of others allows them to reduce their burden. A cell surrounded by neighbors that secrete protective compounds no longer needs to do the same. It can reallocate its limited resources, shifting its internal chemistry toward other functions, and so slowly roles begin to emerge. No cell commands, no cell obeys. But through sheer biochemical necessity, a division of labor forms. The community, if it can be called that, becomes more than the sum of its parts. This division is fragile. If too many cells stop producing a shared resource, the group fails. If too many cheat the system, the environment degrades, and yet time filters the extremes. Clusters that strike a balance persist longer. Those that do not are undone by entropy and scarcity. This is not fairness. It is not justice. It is the quiet arithmetic of survival, cooperation. It turns out does not require intention. It only requires constraint. Some bacteria today still live in such communities, mats, colonies, chains, remnants of these ancient arrangements. Cyanobacteria, for instance, often organize into filamentous strands where some cells fix nitrogen and others photosynthesize. This coordination is not planned, it is inherited. Regulatory circuits activate or repress genes depending on position, nutrient access, or local concentration gradients. The result is an emergent pattern, a tissue without tissue, a body without organs. It is not that each cell knows what the whole needs. Rather, the whole arises because the parts respond locally in predictable ways. Like ripples aligning into waves, form emerges from repetition. In these early cooperative systems, one of the most profound transitions begins: spatial organization of function. Cells stop being interchangeable. Their location starts to matter. A cell on the periphery may be exposed to more light, while one deeper in the cluster may face lower oxygen. Each responds differently, not by command, but by necessity. Over time, gene expression becomes context-dependent. Cells of identical genotype behave differently because their environment varies by microns, and in that variation, complexity gains a foothold. The mystery is not that cooperation occurs, it is that it holds. In a system with no mind, no reward, no punishment, how does selfishness not overwhelm? The answer perhaps lies in the cost of defection. A cell that takes but does not give may benefit for a while, but if its behavior destabilizes the group, it suffers with the collapse. Those lineages that learn chemically, not cognitively, to stabilize their surroundings persist. Those that exploit without limit disappear. Evolution, slow and impartial, selects not for altruism but for compatibility. Traits that promote cohesion survive because cohesion promotes survival. Eventually, molecular signals begin to appear, small molecules secreted into the environment to communicate cell density, stress, or nutrient levels. These are not language, they are not messages, they are gradients of information. Quorum sensing in bacteria, for instance, allows populations to coordinate gene expression once a threshold number of cells is reached. At low density, each cell acts independently. At high density, collective behaviors biofilm formation, bioluminescence, sporulation are triggered. Again, no cell decides, no vote is taken. The system responds because the molecules reach a concentration that tips a switch. Like rain forming from humidity, action emerges from accumulation. These principles, local interaction, emergent behavior, conditional cooperation, become the scaffolding on which multicellular life will later rise. But even in their primitive form, they demonstrate a truth that will echo through all biology. Complexity does not require control. Organization does not require consciousness. Structures form not from command, but from compatibility, from the alignment of internal dynamics with external conditions. Only took a few shared molecules to spark mutual benefit. Only took a local concentration shift to trigger new behavior. Only took the friction of resource scarcity to favor groups that lasted a little longer. In these early clusters, the architecture of bodies is nowhere to be seen. Yet its logic is already whispering through the shared cytoplasmic waters, not yet limbs or nerves or layers, but roles and regions. A choreography without music. Modern multicellularity has evolved many times in animals, plants, fungi, algae, each lineage building on this ancient grammar of coordination. Yet all of them still depend on the same principles: differentiation by context, cooperation through constraint communication via chemical gradients. These are not traits that had to be invented, they are traits that had only to be stabilized. The early cells do not know they are building the future. They simply persist where persistence is possible. Their cooperation is a byproduct, their differentiation an outcome, their organization a side effect of survival. And yet from these small repetitive acts of clustering of specialization, of staying the first whispers of organismal form begin to rise, not suddenly, not clearly, but like a coastline seen from beneath the waves. The early clusters of cells drifting in mats and filaments gradually give way to more persistent associations, not just of proximity, but of position. Cells remain where they are born. They inherit their neighbors. They stop drifting and start belonging. Within these clusters, shapes begin to matter. A layer of cells thickens. Some form surfaces. Others recede into the interior where oxygen is lower, light fainter, and nutrients less fresh. Differentiation sharpens. Position begins to dictate function. It is not design not yet, but a bias, a preference written in the gradients of need and opportunity. Formed slowly and without plan begins to influence function. The geometry of a cluster affects what it can do. A flat sheet offers maximum exposure to light. A tubular form channels flow. A spherical clump preserves interior chemistry. These shapes are not chosen. They emerge from mechanical constraints, from adhesion, from surface tension, from diffusion limits. Cells push against one another, secrete sticky proteins, deform membranes, and in doing so they sculpt the earliest tissues. Inside these primitive bodies, roles stabilize. Peripheral cells become more metabolically active. Inner cells slow down, some even becoming quiescent. Some clusters evolve simple divisions, outer cells for protection, inner cells for reproduction. Already a distinction emerges between soma and germ, between those that support life and those that transmit it. This division is not enforced by a leader. It arises from spatial gradients of signal molecules, nutrients, waste. Cells sense their environment and respond. Over time, those responses are etched into gene regulation, when here behave this way, when there behave another. This rulebook is not written in words, but in conditional expression, in proteins that bind DNA more tightly under certain conditions, in feedback loops that stabilize new states. As structures become more defined, movement becomes possible. Some clusters bend toward light, others contract slightly rippling in response to stimuli. Flagella coordinate. Cytoskeletons align. There is no nervous system yet, but there is coordination born of mechanical and chemical interaction. Signals move as gradients, as waves of ion flow, as the spreading of a protein from one membrane to the next. The multicellular body begins to behave as a single entity, though its mind is nowhere to be found. This is not behavior as we usually define it. It is emergence pattern, response feedback repetition. The constraints of scale now impose new limits. As bodies grow, diffusion alone cannot distribute nutrients or remove waste. Some cells begin to specialize in transport, forming internal canals or open channels. Others begin to sacrifice their growth for the good of the collective, focusing on support or defense. These decisions are not moral. They are biochemical. Cells that stabilize the structure improve the odds of survival for all. Over time, those that act in harmony with neighbors persist. Those that do not are lost. It is one of biology's soft ironies that form does not follow function. It precedes it. The architecture of the body limits what cells can become. A cell deep inside cannot photosynthesize. A cell at the edge cannot retain all nutrients. The geometry shapes the options, and from those options specialization emerges. Complexity then is not built by blueprint, but by the compounding of spatial bias. What begins as simple association becomes a tissue. What begins as a cluster becomes a system, not because anyone intends it, but because form allows function to divide. In modern multicellular organisms, this logic is preserved. During development, the embryo begins as a ball of cells nearly identical. Then, through gradients of morphogenes, chemical signals that diffuse across the embryo cells begin to express different genes. A high concentration here triggers one fate. A lower one there triggers another. This process, known as positional information, echoes the earliest divisions of labor. The embryo is not assembled. It unfolds. It is shaped by gradients, tensions, and the mechanical forces of expanding, folding, and sealing tissues. Even now, with genomes mapped and proteins catalogued, we do not fully understand how form emerges. The cell's internal code offers instructions, but the final structure arises from interaction, from negotiation between inner rules and outer pressures. There are mysteries still unresolved. How did the first stable body plans evolve? Why did certain forms, tube spheres, layers recur across lineages? What made some cellular arrangements robust enough to be inherited while others disappeared? We suspect the answers lie in mechanics, in the laws of surface area and volume, in the physics of membranes and the efficiency of diffusion. But the specifics remain elusive. Complexity leaves fewer fossils than bone. Only took adhesion molecules to hold cells together. Only took regulatory feedback to freeze rolls in place. Only took pressure from the outside and gradients from within to coax shape into meaning. The first multicellular forms are not elaborate. They are not animals or plants. They are sponges, mats, filaments, silent bodies that do not move with intent, but hold their shape against the current. And yet in these forms a profound shift takes place. Life begins to construct itself from more than chemistry. It begins to assemble by architecture. The body, once an accident of clustering, becomes an agent of survival. Its edges sense, its interior regulates, its compartments interact. Function follows the fold, and folds arise from constraint. What once was chaos now has axis surface core. And though the cell remains the unit of life, it is no longer alone. It belongs to something larger, not a self, not yet. But a system that behaves with coherence, responds with coordination, and endures with resilience. In time, these forms will walk fly-root into soil, but for now they float or cling or bend toward light, organizing themselves by where they are and who surrounds them. No voice guides them. No mind plans them. just form finding function again and again until the logic of the body becomes a habit life cannot forget. For billions of years, life unfolds in a world without breath. Cells ferment, reduce, and cycle electrons in quiet chemical loops, never touching the boundless energy locked inside the oxygen they cannot yet make or cannot yet tolerate. In these ancient seas, oxygen is not life, it is danger. A reactive gas, corrosive and volatile, it binds too tightly, rips electrons from delicate structures, turns essential molecules to ash. But somewhere perhaps near the surface in shallower sunlit waters, a different chemistry begins to hum. Cyanobacteria, simple cells bearing pigments that trap sunlight, begin to split water molecules not for thirst, not for cooling, but to harvest electrons. The byproduct is oxygen. At first it bubbles into the water insignificant and scattered. But these cells do not stop. They multiply. They coat surfaces. They bloom. And slowly and visibly oxygen starts to accumulate. At first it binds with dissolved iron, turning the oceans red. The world rusts. Iron falls out of solution, drifts to the seafloor in bands that will one day become stone. Then oxygen reacts with methane, stripping away the gas that once held warmth in the atmosphere. Temperatures drop. Ice creeps toward the equator. The Earth enters one of its first great freezes not because of intention, but because of photosynthesis. Life, by changing its metabolism, begins to change the planet. It does not notice. It cannot. But the consequences deepen. Oxygen, now more than a trace, seeps into every crevice, reacts with every exposed molecule. For anaerobic cells, the ones that thrived without it, this is extinction. Oxygen poisons their enzymes, shreds their membranes, steals the electrons they need to survive. Entire lineages vanish. And yet in this devastation, opportunity appears. Oxygen, for all its violence, carries immense energy. Molecules that burn cleanly, that break apart in oxygen's presence, release far more power than fermentation ever could. Some cells, perhaps already adapted to handling small bursts of oxidative stress, begin to co-opt this reactivity. They evolve pathways to manage the danger. Antioxidant molecules buffer the damage. New enzymes bind oxygen tightly, carefully feeding it into reactions with control. The cell learns not to love oxygen, but to use it. And in doing so, it unlocks a new metabolic world. The rise of oxygen is not swift. It stretches across hundreds of millions of years. The atmosphere shifts in fits and starts. Geologic records show pulses, oxygen rising, falling, stabilizing. Forests do not yet breathe. Lungs do not yet expand. But the air thickens slowly, quietly toward the possibility of future respiration. The cell, meanwhile, continues to refine its inner chemistry. Mitochondria appear not as inventions, but as immigrants. Free-living aerobic bacteria are engulfed by larger host cells not digested but retained. A partnership forms. The engulfed cell provides energy through oxygen metabolism. The host provides shelter nutrients, protection. This symbiosis becomes permanent. The engulfed bacteria lose independence, shed their genome, become mitochondria. And the cell that once fermented now breathes. The irony lingers: what began as waste becomes power. What once poisoned becomes fuel. Oxygen, once feared, is domesticated not in chains but in pathways. The electron transport chain arises step by step, moving electrons along proteins embedded in membranes, pumping protons across gradients, driving the synthesis of ATP. This system is complex yet ancient. All modern eukaryotic life relies on it. It works because oxygen waits at the end, pulling electrons into its grasp, turning potential into motion heat structure. The yield is immense. Cells powered by oxygen can grow larger, move faster, differentiate more deeply. Complexity no longer costs more than it's worth. And yet oxygen remains dangerous. Even now, every breath carries risk. Reactive oxygen species byproducts of this metabolism damage. DNA oxidize, lipids trigger apoptosis. Cells defend themselves with enzymes like superoxide, dismutase, and catalase. These defenses are ancient. Their genetic blueprints trace back to the first oxygen whispers. Life did not master oxygen. It adapted to its violence. It built around it. Even today, organisms that live in low oxygen environments or avoid it entirely are reminders of what once was. Why photosynthesis evolved to split water is still a mystery. Simpler photosynthetic pathways existed before using hydrogen sulfide or other donors. Water is abundant but chemically stable. Breaking it apart requires energy and complexity. Why do it? Perhaps because those that succeeded gained independence from rare resources. Perhaps because once it started, the oxygen-using pathway was so powerful it reshaped everything. We do not know. We see only the echoes in stromatolites, in ancient rocks, in the irreversible shift of Earth's chemistry. Only took a pigment to trap sunlight. Only took an enzyme to split water. Only took the patience of cyanobacteria coating the seafloor for the planet to fill with breath. The atmosphere, once reducing and hostile, becomes oxidative and vibrant. Iron no longer floats. Sulfur cycles change. Methane is driven underground. Evolution does not pause to acknowledge the magnitude of this transition, but it is vast. No other gas has altered life so completely. The Great Oxygenation Event is not an invention. It is a side effect of persistence. Cells that performed oxygenic photosynthesis simply endured, covered more surface, outcompeted others by making their own fuel. They did not seek to change the sky, but the sky changed. Life and planet once separate begin to co-evolve. The biosphere becomes a geological force. Carbon is buried. Oxygen accumulates, climate shifts, ecosystems appear, collapse, rebuild, and the pattern continues. Breath becomes possibility, breath becomes risk, breath becomes rhythm. Today, oxygen supports fire-flight cognition decay. It burns in every cell, stokes the heat of every mammal, rusts every metal surface. Its presence marks the passage of time, aging oxidation, the slow unravelling of structure, and yet we cannot live without it, nor could the first aerobic cell. The partnership with oxygen is not harmonious. It is negotiated, balanced, tolerated. It is a partnership written in electrons and shaped by error, but it holds. And so the Earth turns oxygenated and alive, bearing the signature of those first photosynthetic cells. They left no bones, no cities, no myths, only air, only the gift and burden of breath. In a quiet fold of evolutionary time, some cells begin to draw their genetic material inward, enclosing it behind another membrane separate from the swirl of cytoplasmic reactions. This membrane, double-layered and porous, does not trap the DNA in isolation. It filters access, slows interactions, modulates what passes in and what flows out. This is the origin of the nucleus, not a command center, not a seat of consciousness, but a buffer. Within its bounds, DNA coils and uncoils, folds into territories, loops into contact with regulatory proteins. The chromatin shifts shape depending on the needs of the cell, more open here, more tightly packed there. Transcription becomes selective, no longer the free-for-all of prokaryotic expression, but a controlled response to internal and external cues. This pause, this separation, introduces something life has not yet fully wielded: the ability to wait, to time the act of expression, to separate memory from motion. The nuclear envelope is not an impervious shell. Its pores are complex assemblies of proteins' gates that allow passage of RNA proteins, ions, each screened, each timed. Through these pores, messenger RNA exits the nucleus carrying coded sequences from the genome to the ribosomes in the cytoplasm. Translation happens outside, but the choice of what gets translated and when is now a modulated process. Eukaryotic cells do not just copy their genes, they process them. Introns are removed, exons are joined, regulatory elements bind and unbind, folding the genome into physical shapes that affect what is read and what remains silent. This architecture of regulation introduces new layers of complexity. The nucleus is not simply a container for DNA, it is an arena for choreography, where loops of sequence and clouds of proteins assemble, disassemble, delay. This internal partitioning is not limited to the genome. Other compartments form endoplasmic reticulum, Golgi apparatus vesicles, lysosomes, each wrapped in membrane, each with specialized tasks. The cell becomes a city of compartments, a map of zones and flows where spatial separation allows conflicting chemistries to coexist. Acidic lysosomes digest waste. The endoplasmic reticulum folds proteins. The Golgi tags and sorts them. Mitochondria churn with oxidative gradients. And at the center of this spatial logic sits the nucleus, holding the cell's long-term plan in stasis until conditions justify its release. Why enclose the genome at all? Bacteria thrive without this feature. Their DNA floats in cytoplasm, translated as it is transcribed. Speed and efficiency are high, but what eukaryotes lose in immediacy, they gain in control. The nuclear envelope introduces delay, and in delay flexibility. A gene can be transcribed, its transcript spliced in multiple ways, regulated post-transcriptionally, held in storage, or degraded before it ever becomes protein. This multi-level control opens the door to complex differentiation, to long developmental timelines, to responses that unfold not instantly, but with nuance. Only took a fold in membrane to create the envelope. Only took a series of pores to regulate exchange. Only took the persistence of DNA-binding proteins to scaffold genomes into shapes that remember not just sequence but history. In this way, the nucleus is not just a chamber of storage, it is a chamber of possibility. It holds what the cell might become without forcing it to become it all at once. The presence of the nucleus also changes the shape of the genome itself. Linear chromosomes replace circular ones. Telomeres evolve buffers at the ends of DNA strands that delay the erosion of essential code. New enzymes arise to replicate and repair this expanded, segmented genome. Cell division grows more complex. Mitosis emerges not as an invention but as a necessity. Chromosomes condense, align, are pulled apart by spindle fibers, another layer of choreography ensuring that each daughter cell inherits the full blueprint. This added complexity is not without cost. Errors during mitosis can be catastrophic. But the benefits of modularity of regulatory potential outweigh the risks. Life now has the tools to build in layers. The nucleus also enables a more nuanced relationship between environment and expression. External signals trigger internal cascades, phosphorylation chains, second messengers that ultimately affect nuclear transcription. The cell becomes a sensor, a mediator, between the outside world and the potential locked within its genome. This linkage is not perfect, it is prone to noise, but it is powerful. Cells can now respond differently to the same environment depending on their history, their position, their neighboring signals. Development becomes possible, so does specialization. There are uncertainties still. How exactly did the nucleus evolve? Was it a simple invagination of membrane or a symbiotic relationship with another organism? Did its components assemble gradually or appear abruptly under selective pressure? The fossil record offers no answer. Only the modern eukaryotic cell with its bewildering array of internal structures hints at the story. We reconstruct from homology, from molecular machinery, from genetic parallels. But many details are lost. What is clear is the nucleus did not create complexity, but it enabled it. By slowing the connection between DNA and protein, it allowed a buffer, a stepwise logic, a filter between information and action. And in that gap, evolution found room to build. Even now the nucleus is not static. It pulses. Its envelope dissolves and reforms during division. Its chromatin shifts with age, with stress, with disease. But it endures. It holds. It allows the cell to become many things over time, to pause, to prepare, to store silence as well as sequence. Only took a layer of lipid to change the pace of life. Only took a few delays to open the space for timing pattern differentiation. Only took a sealed room in the middle of the cell for memory to decouple from immediacy. The nucleus is not a mind. It does not think. But it allows thinking to one day emerge. Not yet, not here, but someday. The cell, already a complex gathering of compartments and gradients, begins to welcome guests. It does not plan. This does not set out to merge with another life form, but evolution, like water, often flows where resistance is lowest. Somewhere in the deep past, an ancestral eukaryotic cell engulfs a free-living bacterium, not to consume but to shelter. The bacterium, capable of metabolizing oxygen with efficiency, finds a stable niche inside its host. The host in turn gains access to far more energy than fermentation or glycolysis alone could provide. This is not fusion, it is residence. A quiet agreement shaped not by understanding but by benefit. Over time, what begins as coexistence becomes dependency. The bacterium loses its autonomy, sheds most of its genome, gives up its wall. It divides when the host does lives, when the host lives and dies with it. It becomes a mitochondrion. This moment of endosymbiosis is not loud. It is not marked by a single event. It unfolds slowly across generations, shaped by mutation selection and the quiet logic of necessity. The host cell cannot sustain its size and complexity without a stable energy source. The guest bacterium cannot survive without the host protection, so they remain together, adjusting, refining, entangling. Genes once held by the bacterium begin to migrate into the host nucleus. Protein import pathways evolve to send the translated products back into the mitochondrion. Communication becomes biochemical, precise, irreversible. The partnership hardens into structure. The guest is no longer a guest. It is part of the self. Later, a similar process repeats. Another cell, this one already eukaryotic, engulfs a photosynthetic bacterium, an ancestor of modern cyanobacteria. Once again, the guest survives. Once again, it trades autonomy for safety. Over time, it too becomes internalized, reduced, regulated. It becomes a chloroplast. With it, the host gains the power to harness light directly, to split water, to fix carbon. The result is a lineage that will become plants' algae and countless photosynthetic protists. The world changes again, shaped by the same quiet process, one cell living inside another, not briefly but forever. Symbiosis is not a new concept in biology. Even in prokaryotes, metabolic cooperation abounds. But endosymbiosis is different. It collapses two once independent lives into one, creating an organism with two evolutionary histories. Mitochondria and chloroplasts still retain traces of their origins. They carry circular DNA. They replicate independently. They bear double membranes. These are fossils not of stone but of function, whispers of autonomy, embedded inside every breath and every leaf. Why these partnerships succeeded while others failed remains uncertain. Most engulfed cells are digested or destroyed, but in rare cases the chemistry aligns. The host does not recognize the guest as foreign. The guest does not rupture the host from within. This tolerance, rare and precarious, opens the possibility for long-term integration. And once integration begins, the path forward is one of increasing dependency. Functions are shared, redundancies are lost, genes are relocated. The host cannot remove the guest without collapsing its energy balance. The guest cannot leave without losing the environment it now depends upon. They become together a composite. The implications are profound. With mitochondria, cells can generate enough ATP to support larger genomes, more complex regulation, and greater size. With chloroplasts, cells can feed themselves from sunlight and air. These capabilities reshape not only the cell but the planet. Entire ecosystems arise around these new powers. Oxygen accumulates further. Carbon cycles deepen. Food chains stretch longer. Life diversifies into forms and niches that fermentation alone could never have supported. Only took a single engulfment to change the energy economy of the cell. Only took a failure to digest to begin a partnership that outlasts empires. Only took the passage of time to turn a foreigner into a factory, a guest into a generator. And still mitochondria bear their scars. Their DNA is small, stripped down to essentials. Most of their needs are met by nuclear genes. Their replication is not linked to the cell cycle, but proceeds independently. Some cells contain hundreds of them, others just a few. Their number adjusts with demand, with environment, with stress. They move within the cell fuse and divide, respond to signals they no longer fully understand. Chloroplasts too carry remnants of their independence. Their thylakoid membranes, their internal compartments, their retention of a small genome all speak of a life once lived alone. Even today their functioning is regulated by both nuclear and chloroplast genes, a partnership that must remain synchronized for photosynthesis to proceed. The cell, in managing these organelles, becomes a conductor of internal symbiosis, balancing ancient dialogues within every leaf and every fiber of muscle. There are other cases more recent still unfolding. Certain marine protists have engulfed algae, retained their chloroplasts and photosynthesized for generations. Some insects harbor bacterial endosymbionts that provide essential amino acids or vitamins. These examples suggest that endosymbiosis is not a one-time event, but a recurring possibility, a natural extension of cooperation into intimacy, of interaction into identity. And yet each successful instance is rare. The conditions must be precise, the adjustments must be tolerated, the losses must be survivable. The reward, however, is immense: energy in abundance, light harnessed, function divided and shared, identity rewritten. Symbiosis in slow motion is not a blending, it is a redefinition of self. It blurs the line between individual and collective, between organism and ecosystem. A eukaryotic cell is not one being, it is many-layered, negotiated, integrated. The nucleus and the mitochondrion, the chloroplast and the cytoskeleton, each with their own origin, now coexist as parts of a whole that none of them could have imagined had imagining been possible. Only took a failure to expel. Only took a cell that did not digest. Only took the patience of coexistence to turn two lineages into one. And from that union life grew taller, deeper, more intricate, not because it had to, but because it could. In the depths of a fertilized egg, life begins not with motion but with division, and not with variety but with sameness. Cell after cell, identical, multiplying without pause, without rest, without apparent direction. But slowly as the number increases, the silence begins to change. Genes switch on or off, not randomly, but in response to where each cell is, what it senses, what its neighbors signal. Gradients of molecules, morphogens, diffuse through the growing mass, their concentrations higher at one pole, lower at another. Cells respond to these differences not by thinking, but by unfolding internal programs triggered only under certain conditions. Some genes are activated by high concentrations, others by low. Some require a precise ratio. The genome, once uniformly available, begins to act selectively. The embryo does not yet have form, but it has instruction. and from that instruction pattern begins to whisper into the blank field of identical cells. This process, known as gene regulation, is ancient and pervasive. It begins long before organs, long before tissues. Cells begin to behave differently based on cues that alter what parts of their DNA they can access. Transcription factors bind to promoters and enhancers, creating combinations of permission and prevention. Some genes are read more often. Others are silenced sometimes for the rest of the cell's life. This selective expression gives rise to specialization. A muscle cell does not contain different genes from a neuron. It simply reads a different subset. Identity at the cellular level is not determined by content but by interpretation. The patterns that emerge are not simple. They are recursive, interlocking, self-reinforcing. One gene turns on another which suppresses a third which loops back to stabilize the first. These networks of regulation, like miniature weather systems, settle into stable states, or sometimes chaotic ones. A cell's fate is sealed not by command, but by reaching a point in this network where further change becomes improbable. It commits to a path not with intention, but through biochemical inevitability. Differentiation then is not a decision. It is the outcome of a narrowing field of options. In some systems, these networks form oscillators. Genes activate and suppress each other in loops that create cycles, rhythms that guide when a cell should divide, when it should migrate, when it should pause. These cycles can synchronize across populations of cells, creating tissue-level coordination without a conductor. Hair follicles grow in waves. Limb buds elongate in pulses. The embryo, though without a brain, begins to exhibit rhythm. It is not alive in the way we usually mean, but it is coordinated, timed, and deeply organized. Pattern formation is not limited to internal signals. Cells also use physical forces to shape themselves. They pull on each other, adhere with different strengths, secrete extracellular matrix that stiffens or softens the local environment. The tissue folds, creases, buds. A flat sheet rolls into a tube. A thickened area pushes inward. These physical morphologies feed back into gene expression. Cells under tension may express different proteins than those in compression. The body begins to build itself not just from code but from constraint. Form and function spiral upward together, each informing the other. Why certain patterns recur remains a question. Why stripes and spots appear. Why segmentation arises again and again across phyla. Why limbs grow from specific locations. These are questions partially answered by reaction diffusion models, by positional information theories, by studies in developmental genetics. But behind every explanation is a deeper truth. Pattern is not imposed. It emerges. It is coaxed into being by rules that interact, not dictated from above. One of biology's quiet triumphs is the conservation of these rules. A handful of genes, Hox genes for example, determine body plans across vast evolutionary distances. The same genes that set the head-to-tail axis in a fly help shape the spine of a human. These genes do not specify structure directly. They regulate timing, placement, proportion. Their expression forms gradients. Their activation boundaries map the future landscape of the organism. Only took a few molecules placed just so to ripple outward into anatomy. And yet with all this regularity, variation persists. Small changes in regulatory sequences, in timing and expression thresholds, can shift patterns dramatically. A mutation might delay a gene's activation by an hour, altering limb placement, organ size, or body symmetry. Evolution often acts on these regulatory regions, reshaping old tools into new architectures. It is not the genes themselves that change, but how and when they are used. Development is not static. It is evolution's canvas. Today, developmental biology continues to uncover these deep symmetries and subtle differences. We now know that many traits thought to be unique are built from conserved pathways modified by layers of regulation. The diversity of life's forms arises not from infinite genes, but from infinite ways of using the same ones. A fly wing and a human arm share more in common genetically than their appearances suggest. Only the orchestration differs. There are uncertainties still. What determines when a pattern stabilizes or unravels? Why do some embryos tolerate variation while others collapse from slight disruption? How much of form is encoded and how much is emergent? These questions point toward the boundary between instruction and interaction, between genome and environment, between the written and the shaped. Only took a gradient to tip the balance. Only took a gene turning on in one place but not another. Only took a fold in a field of cells to define an axis, a segment, a limb. The body is not sculpted by hand. It is unfolded by timing, tension, and transcription. Pattern is not a destination, it is a drift. And yet it arrives again and again in fins and feathers, petals and jaws, lungs and gills, always emerging from cells that once were the same. The silence of the undifferentiated becomes the rhythm of becoming, and from that rhythm structure grows, not by will, not by plan, but by the quiet insistence of pattern in a world made of gradients. Within a narrow ribbon of cells, something ancient begins to repeat. Not once, but again and again along a single axis, a rhythm of structure unfolds, not in sound, not in thought, but in tissue. Segments, one after another, each nearly identical to the last, marked by repeating bands of muscle, nerve, gut, and cuticle. In the body of a worm, simplicity becomes sequence. The organism is not a single shape, but a modular stack. This segmentation is not decorative, it is foundational. It creates redundancy, resilience, rhythm. A body built of parts that mirror one another not perfectly, but sufficiently gains a new kind of strength, the ability to elongate, to flex, to move with coordination along its length. The worm's motion is not commanded from a center. It emerges from waves of contraction, one segment after another, tightening, releasing, passing the pulse down the line like a whisper. Segmentation or metamerism arises independently in several lineages, annelids, arthropods, chordates, suggesting that its logic is both useful and accessible. It allows a long body to grow incrementally, to repair damage locally, to specialize parts while preserving the whole. In embryonic development, it often begins with a clock, a molecular oscillator that ticks as cells divide and move. With each tick, a new segment is marked off its boundaries, defined by gene expression cycles and morphogen gradients. Cells at the leading edge of growth interpret time as space. One oscillation becomes one segment. The body is not drawn all at once. It is printed rhythmically from head to tail. This process requires coordination but not central control. The segmentation clock involves genes that inhibit and activate one another in timed loops, notch FGF drifting in and out of synchrony along the embryonic axis. Their products diffuse a degraded signal back. The result is a standing wave of transcriptional activity carving the body into modules. Once established, these segments can become anything. Some remain identical. Others specialize into limbs, wings, ribs, or organs. The initial rhythm sets the scaffold. Later signals determine fate. In the worm, segmentation remains simple. Each unit carries similar structures. The nervous system develops as a chain of ganglia, each segment with its own mini-brain connected to its neighbors. Muscles anchor in repeating blocks. The gut threads through continuous but compartmentalized. This arrangement permits local autonomy with global coordination. If one segment is damaged, the rest may continue. If one contracts, its neighbor responds. The body moves like a ripple. It does not need speed, it needs certainty. There is irony in this architecture. The worm appears simple, yet its development demands exquisite timing and control. Every segment must arise at the right moment with the right boundaries from the same pool of undifferentiated cells. If the clock fails, if the cycle is too fast, too slow, too unstable, the pattern breaks. Segments fuse or fragment. The rhythm dissolves. Precision emerges not from complexity, but from repetition. Why segmentation evolved remains a subject of inquiry. Some suggest it arose to facilitate locomotion. Others argue it allowed for easier genetic and developmental modularity. By repeating structures, evolution could tweak one copy without disrupting the whole. A change in the third segment could yield a new structure, an arm, an antenna, a gill, while the others remained functional. This is not foresight, it is flexibility. Segmental design makes evolution more tolerant of variation. The spine of the worm is not a spine as vertebrates know it. It is soft, unossified, lacking the notochord that will later stiffen other lineages. But it is directional. It defines a head and a tail, a dorsal and a ventral side. It supports bilateral symmetry, the mirrored halves that characterize most animal bodies. From this plan, more complex forms will evolve, but the principle remains rhythm gives rise to structure. Structure allows for motion. Motion enables survival. Incordate segmentation becomes internalized. Somites form along the neural tube blocks of mesoderm that will become vertebrae muscles dermis. The same clock ticks, the same wave passes. Genes that defined worm segments now pattern our own spines. Hox's genes, those ancient position markers, assign identities to each segment cervical, thoracic, lumbar. The vertebrate body is not assembled from scratch. It is sculpted from rhythm, only took a feedback loop to establish a clock. Only took a few repeating genes to divide a body into modules. Only took the patience of development to carve segments into function. The worm sliding silently through soil or sediment does not know it carries the blueprint for complexity. Its segments do not seek limbs, but the potential is there, not because the worm will change, but because the system that makes it allows change. Segmental structures recur across phyla, centipedes, fish, earthworms. Each iteration follows the same quiet rule: repeat what works. Modify what is needed. This is not mindless copying, it is rhythmic adaptation. Evolution listens to the pulse of development, alters the beat, shifts the outcomes. A segment elongates, a pair of limbs appears, a rib forms, a jaw. There are mysteries here still. How did segmentation arise in different lineages? Convergent evolution, or deep homology? Why do some species lose segmentation as adults, even if they display it as embryos? What governs the pace of the segmentation clock, and why does it vary between species? These questions thread through genetics, embryology, paleontology unanswered, but not silent. What remains clear is that the segmented body is a canvas for variation. Its repeated units provide scaffolding. Its rhythm provides timing. Its architecture permits both replication and innovation. The worm in its quiet crawl holds the logic of vertebrates, the seed of legs, the whisper of flight. Not in intention, but in structure. Not in outcome, but in possibility. Only took a pulse to mark a boundary. Only took a delay to space a rhythm. Only took a ribbon of cells listening to the same molecular beat to stretch into a body. And from that body motion, and from motion the search for food shelter mates. The worm does not know it is segmented, but it lives by the rules that segments impose, rules that ripple outward through time, shaping not just worms but whales. From the silent repetition of segments, a new kind of structure begins to extend limbs not as symmetrical redundancies, but as coordinated outgrowths, shaped by gradients, timing, and the whisper-thin instructions of gene expression. A limb is not added, it emerges. It buds from a sheet of cells rising outward into three-dimensional space, not with intent but with inevitability. The location of the bud is not guessed. It is calculated through chemical fields that intersect where axes anterior, posterior, dorsal, ventral, proximal, distal cross at precise coordinates defined by the diffusion of morphogens like fibroblast growth factors, sonic hedgehog proteins, and W&T signals. These gradients do not say build a limb, they say grow here, grow like this, stop there. The rest is interpretation. Cells within the limb bud proliferate not evenly, but according to position. Some remain near the base, others migrate toward the tip. A special region, the apical ectodermal ridge, forms at the outermost edge, maintaining the bud's extension. It secretes signals that prevent differentiation, keeping nearby cells undifferentiated and dividing. As the cells move farther from this ridge, they begin to specialize bone, muscle, tendon, skin, depending on what signals they encounter, how long they were exposed, and in what sequence. It is not a map being followed. It is a timing game. The same gene expressed early might build a shoulder, expressed later a digit. Duration becomes dimension. The limb elongates in layers, its internal structure shaped by more signal zone of polarizing activity, gradients of retinoic acid, carefully choreographed repression and activation. Digits form not as additions but as spaces between areas of cell death. The hand or foot is sculpted by absence. Webbing regresses where programmed cell death proceeds. Where it does not, the web remains. Differences in gene activity, even by minutes or microns, result in claws or fingers, fins or wings. Evolution does not need to invent new genes. It alters the timing, the thresholds, the sensitivity of those already present. Beneath these surface changes, deep homology ties all limbs together. The bones of a bat's wing, a whale's flipper, a horse's hoof, and a human hand all trace back to the same ancestral arrangement. Humerus, radius, ulna, carpals, metacarpals, phalanges repeated, stretched, compressed, split, merged. Their differences are not genetic revolutions. They are developmental recalibrations. Only took a shift in growth rate to lengthen a finger. Only took a delay in cartilage ossification to allow flight. The math of the limb is not complex in code but in sequence, and sequence once altered rewrites form. This modularity allows limbs to be endlessly reshaped. In insects, each leg follows the same basic plan, but variation in segment number and joint position gives rise to walkers, jumpers, diggers, swimmers. In vertebrates, a limb can become a grasping tool, a lever, a wing, or a paddle. The underlying process remains conserved. What changes is the interpretation of the same instruction set. The limb is a developmental theme played in infinite variations. There is no architect, there is no drawing. The plan is distributed across genes and gradients between signals that act locally and networks that stabilize outcomes. Mutations do not act in isolation. They perturb relationships between protein and promoter, between enhancer and repressor, between cell and environment. A single change may have no effect, or it may ripple through the system, altering length number orientation. Most mutations are silent. Some are lethal. A few are transformational. Limbs also demand coordination with the rest of the body. Muscles must connect to bones. Nerves must grow along pre-patterned routes guided by chemical cues. Blood vessels must invade the growing tissue. The whole process unfolds in tight synchrony, guided by feedback from the developing structures themselves. If a limb is removed experimentally, the surrounding tissue often compensates. If two limb buds form too close, they may fuse. If a signal is misregulated, extra digits may appear or none at all. This fragility is not failure, it is responsiveness. Even the left and right limbs are mirrored through a cascade of early symmetry-breaking events. The embryo begins symmetrical, but subtle differences in cilia motion ion transport or transcriptional bias create left-right asymmetry. These early cues influence not just limb placement but organ orientation, vascular branching, neural development. The body is built on a scaffold of symmetry, but it refines itself through asymmetry. There are questions that remain open. How do cells interpret overlapping gradients with such precision? How do genes coordinate timing across millions of cells without a central clock? How did the earliest limbs evolve from fins or from proto-limbs in soft-bodied ancestors? Fossils offer snapshots. Comparative genomics fills in gaps, but the full continuity remains partially obscured, recorded in mechanisms we only partially understand. What is known is this: the limb is not added to the body. It emerges from it as a logical extension of developmental principles already in place. The same gradients that pattern the trunk are repurposed for the bud. The same oscillators that shaped segments now time digits. The system scales, reuses and adapts. Only took a twist in expression to shift a bud forward. Only took a subtle delay to bend the elbow. Only took a repurposing of ancient genes to give a fish the shadow of a hand. The quiet math of limbs is the math of thresholds, delays, feedback loops and gradients. It is not written in numbers but in interactions. The limb does not know it is a limb. It simply follows the local rules, and from them emerges something capable of grasping, walking, flying. The final shape is not inevitable. It is not perfect, but it works. And in biology, what works continues. Limbs in all their variation continue to form each generation, retracing the steps of their own construction, unfolding again the quiet equations of life's architecture, one axis, one gradient, one cell at a time. Life does not leap into complexity. It drifts. Slowly over eons, systems that once operated in isolation begin to intersect, coordinate, depend. What began as a boundary becomes an interface. What began as a signal becomes a conversation. Cells once free-living and self-sufficient embed themselves in bodies that cannot function without each part contributing something the others lack. Differentiation sharpens, lineages specialize, the neuron no longer divides, the red blood cell loses its nucleus, the hepatocyte manages toxins that no other cell encounters. It is not that complexity is sought, it is that redundancy becomes opportunity. The more there is to manage, the more roles there are to fill, and the more roles, the more finely tuned the cooperation must be. This drift is not guided by foresight, but by feedback. What functions persists. What integrates stabilizes. What fails to cooperate fades. Multicellular organisms become mosaics of context cells organized not only by type, but by location, signal exposure, developmental history. One genome, thousands of phenotypes. A skin cell and a retinal photoreceptor share the same DNA, but the former guards and renews, while the latter translates photons into perception. Their differences lie in which genes are silent, which are transcribed, which proteins modify, which histones, in which places at which times. The language of complexity is not genetic alone, it is epigenetic, proteomic, interactive, environmental. A body becomes a layered text, read differently by every cell within it. As organisms grow larger and more sophisticated, communication becomes paramount. Signals travel not just by diffusion, but along axons through blood across synapses. Hormones coordinate distant tissues. Neurotransmitters time rapid responses. Feedback loops between organs adjust blood pressure, temperature, hunger, stress. Each level of complexity nests within the next molecular reaction, shapes cellular behavior. Cellular behavior builds tissues. Tissues create organs. Organs support the organism. The whole remains coherent only because every part remains responsive. The heartbeat accelerates with fear. The immune system activates with injury. Muscles contract, lungs expand, skin sweats, all without conscious command. Some cells give up autonomy entirely. Stem cells divide, but their progeny do not. They become terminal, locked into roles they will never leave. A neuron may live 100 years, but will never replicate again. Complexity demands sacrifice. Efficiency demands specialization. And with specialization comes dependence. No cell survives long outside its context. Remove it from its tissue and it dies or reverts. Complexity binds components together not with force, but with mutual necessity. Development once a matter of patterning becomes a matter of orchestration. Timing becomes as critical as form. A gene expressed too early yields disarray. Too late, and function is lost. Organisms evolve not just new structures, but new sequences of emergence. The eye, the heart, the brain, none appear whole. They arise from folds, fusions, migrations, regressions. The embryo becomes a history of itself, replayed each generation with minor edits, waiting for the point where function catches up with form. The nervous system grows as an interface between body and world. It is not a command center, but a distributed network of sensors and circuits. Reflexes evolve first simple loops between stimulus and response. Then layers are added ganglia, then cords, then brains. Each stage allows the organism to respond not just to the present, but to patterns, to change, to memory. Complexity crosses into cognition, not at a threshold, but along a continuum. It is not a switch, it is a slope. At some point, behavior becomes flexible, learning appears. Trial and error is not just tolerated, but retained. Organisms begin to model their environment, however simply. They remember where food was found, where danger arose. Brains become prediction machines. The future, once irrelevant, becomes a consideration. This change is profound, but it builds upon everything that came before. Gradients, patterns, segmentation, specialization, feedback. No part of complexity is disconnected from its origins. The same rules that shaped a worm's segments underlie a bird's wing, a child's spine, a thought. There are costs. Complex organisms reproduce more slowly. They require more energy, more time to develop, more care to survive. Their failure points increase. A misfolded protein, a mistimed signal, a misplaced cell division can have cascading consequences. But the benefits, flexibility, adaptability, responsiveness open new ecological niches. Complexity expands the map of possibility, and yet complexity is not the goal of life. It is not inevitable. Most life remains simple. Bacteria, archaea, single-celled eukaryotes, they thrive, adapt, colonize extremes. They mutate, recombine, form biofilms, swap genes across species. They are not waiting to become something else. They are already complete. Complexity is not better. It is different. It is one of many solutions to the problem of persistence. Still, in some lineages, complexity becomes embedded. It cannot be undone without collapse. A mammal cannot shed its tissues and return to the sea as a bacterium. Its interdependencies are too deep, its body too layered. Evolution now proceeds not by discarding parts, but by refining the systems already in place, adjusting regulation, reshaping anatomy, shifting behaviors. The genome does not grow much, but the organism does. Not just in size, but in responsiveness, in learning, in social interaction, in memory and anticipation. Only took a few feedback loops to stabilize coordination. Only took a shared circulation to link organs. Only took a folding of tissues to house thought. The drift toward complexity is not linear. It branches, stalls, reverses. But in the long view it leaves a record not of triumph, but of experimentation. Organisms that solve their environment persist. Those that integrate survive longer, and those that organize slowly over time become something we call complex. Complexity is not a crown. It is a fabric woven from repetition and variation, layered with time, held together by rules that care nothing for purpose. Life does not aim for intelligence, for beauty, for meaning. It aims for balance, for resilience, for the continuation of what continues. And in some places, for reasons still partly hidden, that balance unfolds into minds that wonder why it happened at all. The cell never stops. Even now, as night drapes itself across the world, as the body sinks into stillness, every cell persists in its quiet rhythm. Membranes flex, proteins fold and unfold, mitochondria hum with gradients that power every breath, every heartbeat. Somewhere deep in the nucleus, chromatin loosens just enough to allow a strand to be transcribed, then tightens again, conserving energy, preserving memory. Complexity does not pause, but it slows, and in this slowing, in this deep and layered quiet life, becomes something softer. Not progress, not motion, but endurance. The body rests, yet it is never idle. Sleep itself is orchestrated by ancient loops, circadian regulators, that tick in time with light, temperature, and silence. Hormones rise and fall. Neurons fire in low, deliberate pulses. The immune system patrols. Cells renew. Tissues heal. Even in sleep form is maintained identity, preserved boundaries held. The logic of organization does not stop when consciousness fades. It continues patiently without need for attention. Perhaps this is the lesson of life's long unfolding, that order does not require direction. That harmony can arise from repetition. That meaning, if it exists, lives not in sudden change, but in the quiet continuity of processes that began long before we named them. From membrane to multicellularity, from gradient to gesture, from reaction to reflection, life organizes itself not for a reason, but because the conditions allow it. And when those conditions persist even for a moment longer, life does too. The story continues. But for now, let it drift. Let the pulse of molecules fade into the hush of sleep. Let the mind rest in the knowledge that beneath every thought, every breath, every dream, the quiet order of life still unfolds, patient and unseen,